first overview of it. All right, so chapter 7 is cellular respiration. Basically, how do cells harvest and create energy so they can carry out all the life processes that they need to. So a little bit of a background introduction here. Plants, algae, some bacteria create or make their own food through photosynthesis. They harvest sunlight and create. It's actually kind of cool. You take light and an air molecule, in this case carbon dioxide, and you literally turn it into a chemical form, sugar. So we call these folks, plants, algae, bacteria that can do this, autotrophs. They're self-feeders. They make their own food. But then on the flip side, they provide all the energy in an ecosystem. We have to eat that in order to get the energy. So then heterotrophs. Oh, I'm sorry. Thank you. My bad. Okay, it's really slow. It's like, come on.
Oh.
You can see it flashing.
Seriously. Zoom. Oh my gosh, it's going to take forever. Oh, wrong line. It's like a 3-second delay.
5 seconds.
Okay.
Not close enough.
No, we'll get there.
Oh, it's still going.
Come on.
Oh,
too much.
Hours.
Okay, just a little bit. All right. I think we're okay. Okay, so anyways, heterotrophs are organisms that eat. The autotrophs are the producers, so they're fed by others. They have to eat in order to get their energy. 95% of the organisms live on organic compounds that the autotrophs or producers produce. And so this whole process of cellular respiration, energy is harvested through the oxidation of organic compounds. So in this case, basically it's a combustion reaction because your byproducts are oxygen and water vapor, right? So let me write out the cellular respiration equation. C6H12O6 plus 6 molecules of oxygen yield 6 molecules of CO2 plus 6 molecules of water. This is a combustion reaction. You have a hydrocarbon, okay? I'm just going to hydrocarbon plus oxygen, okay?
If it's just oxygen, why did you do O2?
Oxygen gas,
Oh,
okay? And you're creating energy. I should put energy in this equation. Carbon dioxide and water vapor. This is a combustion reaction. I could take like ethanol or any type of alcohol, which is a hydrocarbon, expose it to some oxygen, and boom, get a reaction, like big boom. Now, for us, this doesn't happen. You don't explode. So what the process does is it breaks it down into tiny little steps so your cells don't die. Because if your cells were to break down glucose all at once, boom. So it does it slowly. I'm just going to cleave off a carbon, cleave off another carbon, cleave off another carbon. It just keeps going and going and going. So cells oxidize organic compounds to drive this metabolism. So we've talked about the macromolecules, fats, carbohydrates, proteins, how they're rich in these CH as well as CO bonds. So that's why, carbs, we use it as a main energy source, but fats have even more of these bonds. So they actually carry more energy. And so the whole process of cellular respiration is extracting energy and breaking it down into these stages. So we're going to talk about enzymes and how they break down these large molecules into smaller ones, how they dismantle and cleave off certain CH bonds at each little stage and just kind of slowly metabolize or break down glucose into, CO2 and water vapor. So we're breaking down the molecules. However, there's a common feature here. You're going to follow electrons. Okay, so every time that a bond is broken or formed, electrons are being transferred. So one thing to keep in mind during this whole entire process is where did the electrons go? And then over here we have our dehydrogenations, which means the electrons are lost, but they're also kind of picked up with protons. So not only are we going to follow the fates of electrons, but we're also going to follow the fates of protons and how these two play a super important role in how ATP is made. So cellular respiration is all about breaking down glucose. Remember with redox reactions, some of you got that question wrong on the exam. Something about, if electron is reduced, I don't know, it was something like that. And then it's like, what does that mean basically? So if you are reduced, do you gain or lose electrons? You gain. Oil rig. Reduce in gain of electrons. Okay, so some of you got that backwards on that multiple choice. So we're going to, like I said, follow the flow of energy and electrons as it's, bonds are being rearranged. Another energy molecule or electron carrier, I should say, NAD+. You're going to see different forms of NAD+. So in animals, it's NAD+ for cellular respiration. In plants, it's NADPH. They're very similar cofactors that are involved in the redox reactions involved. So NAD+ will accept electrons. So a lot of times we call NAD+ an electron carrier as well as a proton to form that NADH. So this means that no electron, no proton, and then once it picks up an electron and a proton, now it's NADPH. So lots of different little enzymes. This is just one that you need to know. There are dozens of redox reactions. Dozens. In glycolysis alone, there are 10. And you will have to memorize the 10 intermediate molecules. So after an enzyme does, cleaves off this or rearrange that, what's the new molecule called? Like, that's your quiz. Basically, intermediate molecules in glycolysis. So dozens of redox reactions where NAD+ is an electron carrier. So you're going to create several NADPH molecules. Every time an electron is transferred, energy has to be released in order to make ATP to form those chemical bonds. But at the end of the process, you're going to have so many of these electrons, and they're going to help create ATP. Also, with the fate of electrons, it kind of helps you figure out if it's going to be an aerobic respiration or anaerobic respiration. So oxygen, in the process of cellular respiration, if you have oxygen, oxygen acts as an electron acceptor. So if oxygen is present, it will be aerobic respiration. However, if there is no oxygen, now it's going to find a different electron acceptor, and we'll talk about different forms of that, but like lactate acid is one. We call that anaerobic respiration, so no oxygen present. And if there's no oxygen present, you usually end up in a process of fermentation. And we'll talk about the two different types of fermentation, alcoholic and lactic acid. This is where I get all the animations. Okay, so remember in redox reactions, one molecule will lose an electron and give it to another, as well as transfer in a proton. So now this guy, he lost the electron, he has become oxidized. Notice that he has a positive charge, and this guy gained the electron, so he is negative. So you can think of sugar with oxygen, yielding carbon dioxide, water, and ATP. One of these guys, they're going to be oxidized and reduced, and you'll create redox reaction here. Yeah, lots of tiny little animations.
Yeah,
Okay.
so that process is cellular respiration, not photosynthesis.
This is cellular respiration. Yep. So sugar and sugar and oxygen. That's what you start with. So burning carbohydrates. There's a little difference between catabolism of carbs in a cell and burning wood in a fireplace, because wood is made of cellulose, again, another macromolecule of carbs. So the reactants are carbs and oxygen. The products are carbon dioxide, water, and energy. And that energy is usually in the form of heat as well as ATP. To do this entire process, this is the amount in free energy. So if you want to break and form bonds, it's going to be either minus 686 kilocalories or positive 686. But the same amounts released whether glucose is catabolized or burned, which means broken down. Okay. Does anyone remember the opposite of catabolize? Because like this was cannibalism, breaking down. What was the opposite of building up? Animalism.
Oh my god.
Yes,
I was
it's
gonna say
animalism.
animal.
Yep.
I was
So
gonna say-
photosynthesis is like the opposite of it because you're making glucose. Okay, taking tiny little parts and you're making a huge molecule. So photosynthesis is anabolism. Anabolism.
Yeah, because like anabolic steroids.
Anabolic. Thank you. Yes, anabolic. That's the word.
Well, I thought of that and I was like, anabolic steroids. Does that make sense? I decided no.
All right, so electron carriers play a critical role in this entire process. Eventually what you're trying to do, and I know I'm being super redundant, you're taking glucose and you're breaking it down into 6 molecules of carbon dioxide. Okay. If electrons were sent directly to oxygen, the reaction would be combustion and your cells would burst into flames. But don't worry, that doesn't happen. What happens instead is that these electrons get transferred to an immediate or intermediate electron carrier, and it's used for another little process, and then eventually it gets to oxygen. So, yeah. Thank God that doesn't happen. Okay, there are many different forms of electron carriers, and so we'll see them. NAD plus is just one of them. There's also FAD plus, so that's the other one you'll need to know. But the common feature of all carriers is that they can be reversibly oxidized and reduced. Basically, they're saying you can go from NAD plus to NADH and back. Or FAD, I think it's just FAD plus, to FADH and back.
But you said it's NADPH
In plants. Yep, so this would be in cellular respiration, and then in photosynthesis, whoa, what was that? That was an R. Okay, NAD P plus to NADPH. And I don't remember the second electron carrier offhand right now. But yeah, and back. That reverse here. Okay. Some other energy molecules. Or sorry, my bad. No, electron carriers. Looking at the structure of it. So it's made of 2 nucleotides. I don't think I quiz you on the structure of it, but if you're curious, this is NAD plus, okay? And you can see that it's got Na, it's got some nicoantamide adenine, and then dinucleotide. So if you just have one, it's a monophosphate. If it's just, without this part, it's adenine. But the function of each nucleotide, obviously, enzymes recognize. So there's like motifs. They recognize the AMP motif of it. And then this is the part that readily accepts those electrons. That's just like fun fact. I don't think I quiz you on that at all. Okay. Lots of stages here. So next Monday, we're going to go into glycolysis. I think it's next Monday. Actually, I think we might talk about glycolysis today. And then we can review. Okay. But we'll start with glycolysis. And if you look at the prefix glycolysis, glyco is sugar and lysis means split. So the splitting of sugar. And then we'll talk about what products are made from that. And then how those products fit into the Krebs cycle. Eventually, with glycolysis and Krebs cycle, you're generating electron carriers, NAD plus. So then these electron carriers will go to the electron transport chain, which is in the mitochondria. And when they get there, they energize some of these protein channels that help hydrogen flow across, producing a gradient. And then we're going to use that gradient to create ATP. Okay. So here's a diagram showing the comparison between combustion and respiration. As you can see, they're very similar. You got some type of fuel, in this case carbohydrates, wood, in this case sugar, okay, twist cone. We add a little bit of oxygen and something to kind of jumpstart it together. There's oxygen jumpstart together with some enzymes, and you're generating carbon dioxide, water, heat, ATP, but they should really, oh, that's right there. Carbon dioxide, water, and heat. There it is.
So really, since wood's made out of all these carbohydrates, can you just eat wood and get energy from it, or what?
It's because it has cellulose, and your body can't break down cellulose. So,
Oh,
yeah.
that's too bad.
Let's just cut out the cell.
Okay, so remember ATP is the energy currency in cells. It's responsible for cell movement, pulling apart chromosomes, movement of organelles, basically everything that the cell needs to do. It is, they drive endergonic reactions. So endergonic means it absorbs energy. Remember when you cleave off a phosphate, it's, minus 7.3 kilocalories. And then you need 7.3 kilocalories to create ATP again.
So 7.3 kilocalories, that's only with the phosphate, or?
Yep. Like when you take off that last phosphate. So, oxidation of glucose in summary. Like I said, we're going to talk about how ATP is made. And there's two ways ATP can be made. It can be made through substrate level phosphorylation or oxidative phosphorylation. In substrate level phosphorylation, basically you're going to transfer a phosphate group to ADP and you make ATP. Substrate level phosphorylation only occurs in glycolysis. As you can see, you only make 2 ATP molecules. Now, that's not fast enough for your cells, because your cells, you only have a supply of seconds before your body has gone through all the ATP that it has. So it has to find another way to make ATP much faster. So oxidative phosphorylation means that we're going to use a proton gradient, H plus gradient, built up by the electrons that were transferred to the ETC, and these protons are going to flow through an enzyme called ATP synthase and generate a metric buttload of ATP. Okay, so a lot of times you see these two work together, but you get a lot of ATP here and not so much here. I always like to mention that in glycolysis, like your body is always going to do glycolysis, but oxidative phosphorylation, you need oxygen because that's your final electron carrier. So if you don't have oxygen in your cells, like let's say you ran 3.1 miles and you are just like dead, okay, because you ran super hard, it depleted your oxygen levels. I mean, you still have oxygen, but it's not a lot. Well, now your body's going to start going through glycolysis, which will lead to fermentation because you don't have oxygen to pick up those final electrons. So that's why you get, super sore after you push yourself, or you just get a little bit slower at the end of a race because your body is now just doing substrate level and not as much oxidative phosphorylation. So. Okay, we're going to break down all of these series into 4 stages. So we'll talk about how we're going to capture energy by substrate level phosphorylation through glycolysis. There's no oxygen required. Okay, your cells will always do glycolysis, but then it's like a little flow chart. Is there oxygen present or is there not? So the next 3 stages carry out aerobic respiration, which means what? Aerobic? There is oxygen present, and so this is kind of like the flow chart here. Cells always do glycolysis, but then notice we're going to generate some electron carriers. We're also going to generate a molecule of pyruvate, actually 2 molecules of pyruvate, and then everything travels to the mitochondria. So where does glycolysis happen?
Mitochondria. No.
The cytosol.
In the cytosol or the cytoplasm. I would accept either or for that. So this happens in the cytoplasm of your cells. But the mitochondria, that's where these aerobic processes take place. So pyruvate is going to go to the Krebs cycle and get rearranged. I'm going to generate some more ATP, not a lot, it's still substrate level. But then the electrons that are formed from glycolysis and Krebs cycle go to the ETC, and this is where the oxidative phosphorylation occurs. And you notice that the ATP little explosion is a lot bigger compared to these two. So this is where we're gonna generate a bunch of ATP, and
So somehow
These
it's
are just
getting
some
way more
byproducts.
ATP than it's putting in. So wait, wouldn't your body then? Oh, you're using something. Never mind.
yeah, and you're gonna find that in some of the processes, your body has to burn a little bit of ATP,
I
then
was
make
gonna say,
some
wouldn't
more
it just
ATP.
keep getting? You get like an overload in abundance, but then I forgot I did. Yeah.
So ATP central role. Oh, why did it go back? I'm like, I've already said this. Now I'm going backwards. Ugh. All right, stage 1, glycolysis. 10-reaction biochemical pathway. We do generate ATP through substrate level. There are enzymes in the cytoplasm of the cell. There's no membrane or organelles. It just happens in the cytoplasm because there's enzymes there. Your products at the end of glycolysis, you will create 2 ATP molecules. Actually, you have to burn 2, but you make 4. So your net is 2. So
Basically, they're just double.
4 minus 2 is 2. Because this is what we burned. This is what we made. Here's your net. Oh boy. Other products. 4 electrons, and they will be carried out by NADH, and they will travel to the mitochondria, to the ETC. You also, at the end of glycolysis, get 2 energy-rich pyruvate molecules. Pyruvate is a 3-carbon molecule. How many carbons are in glucose?
2.
6. Okay, so glucose is 6 carbons. Basically, you're splitting it into 3-carbon molecules. Oh my god, I can't write today. It's gonna be bad. All right. So 1 molecule of glucose makes 2 molecules of pyruvate. Questions on pyruvate? 6 to 3. As a result, you get 2. Okay, 4 electrons. That's just a simple number. Okay, and then 2 ATP. You burn 2, you make 4. Your net is 2. Okay. So that's glycolysis. How many molecules of pyruvate?
2.
2. How many carbons are in pyruvate?
3.
3. Okay.
So it makes 2.
2 is.
How many
2
ATP
seconds.
molecules are made net?
2.
2. How many does it make?
4.
How many did you burn?
2.
Okay. How many electrons?
4.
4.
You're on a hot streak.
Okay, so then you're going to go to pyruvate oxidation. So this is where it enters the mitochondria. Pyruvate gets converted into carbon dioxide and a 2-carbon molecule called acetyl-CoA. So pyruvate, how many carbons?
3.
3 carbons. And now I'm going to make 1 molecule of CO2 where there's 1 carbon and a 2-carbon molecule called acetyl-CoA. So I'm not making these things up. It's just things are just getting modified and rearranged. So the products for just 1 pyruvate molecule, acetyl-CoA, and a molecule of NAD plus, because of this cleave-in. So I've generated another electron carrier, and I really should add CO2 as another product of
But
this.
isn't that the NAD? Wait, wasn't the CO2 is what changing the NAD plus or no?
Oh, no, okay. All right, pyruvate. 3 carbons, right? What's that?
Something like baseball.
It's only 5 minutes.
So weird that you're just a shadow on them.
Yeah.
Can't even see.
Okay, so 3-carbon pyruvate. I cleave off 1 to create CO2 and acetyl-CoA, but when you break bonds, you're also generating NADH. Yeah. So I really should have another little bullet there of NADH. Okay, so that's just this process, pyruvate oxidation. Okay, and I kind of just wrote it here, but 3-carbon pyruvate broken down into a molecule of CO2, NADH, which will be carried to the ETC and acetyl-CoA. Stage 3 is the Krebs cycle. Okay, so this was stage 1. Here's stage 2, this process. Here's stage 3, the Krebs cycle. So now we're going to take acetyl-CoA, 2-carbon molecule, and it's going to go through 9 reactions. So then when you take your Krebs cycle quiz, you'll have to memorize 9 reactions. So we call this the Krebs cycle. The products for just one acetyl-CoA are going to generate ATP and then lots of electron carriers, just like lots. Like every time it goes through a reaction, you're generating FAD or NADH, like a lot.
But if reduction is in gain, then why is it losing electrons when this is happening?
No, reduction means gain, so they're picking up.
Although I thought you meant, oh, okay. I was thinking like because the thing itself is losing the electrons, but it's because.
Oh no, but it's not. Okay, I
Then
see
you
what you're
need
saying.
plus.
Yes. So acetyl-CoA would be oxidized, and then this is reduced. There we go. Yeah, I see. That's a good question. Yeah. Whew. So this is just for one acetyl-CoA, but how many acetyl-CoA molecules did we create before?
One.
No, it's 2.
It's 2.
Because it was one of the COs.
Yeah, because like pyruvate, you have 2 molecules of pyruvate, and so both molecules get converted into CO2 and acetyl-CoA. Yeah. So overall, really, it's 2 and then a crap ton of electron carriers. So there's the Krebs cycle. And again, these electron carriers are traveling to the ETC, and we make 2 ATP overall. And then the 4th stage will be the ETC. So as you can see, I'm going to spread this chapter out. Like, I think I'm going to touch on glycolysis today. We'll just review it Monday, and you can study for your quiz on Tuesday. But, you know, glycolysis will be one day. And then the next day, we'll talk about the Krebs. Then the next day, oh, sorry, no, I got to back up. Pyruvate oxidation, and the Krebs cycle, and then the ETC. So we'll break it down day by day because it's pretty complex here. But anyways, stage 4 is the ETC and osmosis, basically generating a crap ton of ATP. So energetic electrons carried by those electron carriers transferred to the ETC, where that energy from those electrons is used to pump hydrogen across the membrane. So I'm just going to do a super, super rough sketch of this. So there's this membrane, all right, and this is a channel, we'll say, and there's more membranes. And there's another protein channel, okay? So basically what's going to happen is you have hydrogens on this side, and then your electrons come in and they energize these protein channels, causing hydrogen to move across. So now all these hydrogens are over here. And now I have a high concentration of H plus on this side and low on the inside. And then further down the ETC, there's this crazy enzyme molecule called ATP synthase, and it's crazy looking. Let's see if I can draw it, but it literally looks like a wind turbine. I don't know, something like that. And these hydrogen come in, and when they get to the turbine, it literally spins, and it takes a phosphate.
This is Edie.
Yep, taken out.
Can you send us or send me my test score when you get it graded?
Yep, I can text you.
Thank you.
You're welcome. So then these hydrogens come in and they spin the turbine, and then ADP plus phosphate, it like smacks these 2 molecules together. And what do you get when you take ADP plus P? I had 2 phosphates, and now I'm adding one.
ADPH.
ATP.
Oh yeah, ducks die.
Yeah. It literally looks like it's a wind turbine, like creating energy, like, you know. electricity. But
Maybe
in this
that's why we
case,
modeled
ATP.
the real wind turbines off of it.
Yeah, so we call this process where the protons flow through and create a bunch of ATP, chemiosmosis. Oh, I had a diagram on the next slide. Well,
Literally just the same as what you're doing.
yeah, but anyways, I'll just talk about it again. So these are all electron carriers, and they're dumping off these electrons, energizing these proton pumps that are made of proteins, and they're flowing to the outside here. So now I have a high proton concentration, and then it will go through an ATP synthase, which is a crappy diagram because really there should be a turbine down here, but whatever. ADP plus P, ATP. And then these protons, just start all over again. Okay, that's the overview.
So
You guys-
is there a reason? Can you go back one more?
Sure.
Is there a reason that this whole time you've had the protons labeled as H plus?
Hydrogen is basically just a proton because there's no neutrons
Okay.
and there's no electron. Well, there's one electron. Just focusing it on.
All right. Just because I was confused, because every single thing was like you were pointing to like protons, and you point at H plus, and I was like, wait.
H plus is, yeah, H plus is protons.
Okay. I was thinking it was T. Okay, that's
Okay, here we go. Going into glycolysis. Yeah, I think there's only, we'll see. We'll see how far we get.
give or take.
So glycolysis means splitting of glucose, okay? And so the first half of glycolysis, the first 5 reactions, we're basically taking a molecule of glucose and we're going to create 2 molecules of a 3-carbon compound, which is called G3P, or glyceraldehyde 3-phosphates. And the first 5 requires a little bit of ATP. So this is where we're going to burn some ATP. So this entire process is endergonic. And then the back half, the last 5, are exergonic. So, step A, the glucose priming. Basically, you take your glucose and you create a 2 3-carbon molecule. We burn 2 ATP in the process. Then these 2 3-carbon molecules get rearranged and cleaved a little bit more and rearranged. But eventually, you get to that G3P. So this is the steps. Okay, this is what you'll have to know. Actually, I'll box what you need to know. Basically, you have to know the names. So, glucose gets taken in by an enzyme, hexokinase. So you don't need to know enzymes, okay? But the enzymes may help you out. Hexokinase, burn in 1 molecule of ATP, and you get glucose 6-phosphates. So here's what glucose looks like before. And then notice that we took off a phosphate. So we went from ATP to ADP. Well, they took that phosphate and they just stuck it on the 6-carbon of glucose. So that's why they call it glucose 6-phosphate. Okay. When I was in college, not only did we have to know these, we had to draw them. So you just need to know the names, and you'll see like the molecules and the enzymes. All right, now this intermediate molecule, glucose 6-phosphate, gets rearranged by, wow, that's another enzyme, phosphoglucose isomerase, to become fructose 6-phosphate. Now notice that we didn't add any phosphates, and this is just a rearrangement. So we went from glucose to fructose. So remember when we talked about how these guys have the same chemical formula, but the position of a certain, I don't know if it was an OH or an H, or maybe it was an O, has been moved, okay, rearranged. Then it hits up another enzyme, phosphofructokinase. But we're going to use some ATP here. So if I'm using ATP, that means I'm probably going to add a phosphate somewhere, because now it's just ADP. Where'd that phosphate go? Well, it went on to this fructose 6-phosphate to become fructose 1,6-bisphosphate. So not only is there a phosphate on the 6-carbon, but as well as the 1. So that's why these names, they actually have a purpose. So like, if we memorize the names and we have to draw it, we're like, okay, carbon number 1, that's where that other phosphate went. So here you can see the 2 phosphates. the bisphosphate. Now this is where it gets a little iffy here, okay? Eventually it's going to get to G3P, okay? But it can become another intermediate molecule that's a little bit unstable and then become G3P. So I think on your quiz I give you this. You just need to know G3P, okay? So that's why there's reactions 4 and 5. There's another enzyme, intermediate, then rearranges it to finally G3P. So here's G3P, and as you can see, we have taken our glucose molecule, nice little 5 ring with the 2 carbons there, and we've cleaved it into 3. That's why it's G3P, because there's just 3 carbons now. All right. Now notice that these 2 are the same. Well, that's because I've created 2 molecules of G3P. So then they're like, okay, here's 1 molecule of G3P, here's the fate of the other G3P, and they have the same fates. Questions on that? So like, here's 1 G3P molecule, here's the other G3P molecule. So I've taken glucose and they just like, literally split it right down the middle. We good there?
I think so.
You think so? Okay, I don't know. All right. So, hits up another enzyme, triose phosphate dehydrogenase, and this is where we get our first electron carrier. Okay, NAD plus to NADH, and we get 1,3-bis phosphoglycerate. In that case, 2 molecules of that.
So when
Fancy words.
so when NAD plus goes to NADH, does that mean it lost an electron?
It gained an electron.
Yeah.
50-50 chance, of course.
Okay. Now this is just the first 5. This is not glycolysis. We just completed the endergonic reactions. So I kind of already talked about it, so this will be review. But anyways, ATP synthesized by substrate level. So the ATP that we're going to make in the last half, substrate level phosphorylation. And so we're going to generate some ATP, kind of get a net ATP gain here. But step 3 is the oxidation. We've generated some electron carriers. We're going to add a molecule of phosphate to G3P to get to that 1,3-bis phosphoglycerate. And eventually this phosphate will combine with ATP to form one of those molecules of ATP. So those phosphates that are on, well, they're going to come off again and create ATP. And then step D, generate ATP. So 4 reactions convert 1,3-bis phosphoglycerate into pyruvate. And our net yield for ATP will be 2. So here's the back half. Okay. So this is my 1,3-bis phosphoglycerate. You can see that I have my phosphates attached. Okay. Oh, they didn't draw the pictures on this side, so they're over here. But cleave off the phosphates to make ATP. Now I'm at G3phosphoglycerate. Going through another set of enzymes. We're going to, again, rearrange the phosphates, eventually creating some more ATP, but 2-phosphoglycerate or 2-PG, 2-phosphophenyl pyruvate, and then pyruvate. Because that's the end product of glycolysis. 2 molecules of pyruvate. Things I want to point out. So notice this enzyme, phosphoglycerol, and then it goes kinase, phosphoglycer, and then a different ending. So the enzymes may help you with what the product will be. So phosphoglycerol mutase. So I don't know how you want to figure out the difference between the 2. From going from 3 to 2, I'm sure you could come up with a way. But then pyruvate kinase, I sure hope you guys can put pyruvate at the very end. Okay.
So would that be 1 or 2 points?
Each one of those is a point.
Alright, so 2 points we got for sure.
So could you just write like BPG and
Yes.
3PG?
Absolutely. So essentially,
BPG, 3PG, 2PG.
essentially what it comes down to is people cram, and then that's the way it is. But,
What do you mean cram?
like, oh crap, glycolysis quiz. Okay, BPG to 3PG, and because that sounds like this, and I don't know, they just like come up with these word associations, and
Oh, we're pretty good at that.
then
We're pretty good.
whatever works for you. So brief history of glycolysis. It does generate a small amount of ATP. Your cells do it all the time. It was the primary way that organisms generated ATP in the past. Remember we talked about pathways and how they evolved? They go backwards. So I think I had like a diagram that was like A turns into B, B turns into C, and then D. Well, all of a sudden, let's say we run out of C. So then they had to find another molecule to make C and to carry on the process. And so this is why they think glycolysis has so many steps, is it just kept evolving backwards. We ran out of this molecule. What can we use to make that molecule? Oh, we're now out of that one. What can we use? Okay, so I will review glycolysis on Monday, glycolysis quiz Tuesday. You'll get guided notes.
Students look through the lost and found, it will be gone by Friday. Also, with the